So, a big warm welcome. Very grateful for everybody who's here now and who's watching this later down the track. My name is Sharon Whiteman. I'm the president of the Lyme Disease Association of Australia, and I'm a Lyme patient. I'm a retired ICU nurse, and I'm very passionate about justice and truth and progressing science and health science as well as tick science in Australia. So I started in 2009 with the charity, and in 2026, we're actually further behind them when we started in regards to access to care for people who've had a bad encounter with ticks. And we're very privileged, really privileged to partner with Dr. Mackenzie Kwak. I'm just going to give you a little bit about his background. He's a parasitologist and disease ecologist whose work spans Australia, Southeast Asia, and Japan. He's based in Japan, where he studies ticks, fleas, and mites, exploring how these ectoparasites interact with their hosts and their environments. His research has helped uncover new tick species, including one that parasitizes endangered mountain pygmy possum, and a map where different species occur across varied regions. He has also advanced our understanding on how changes in season, climate, and wildlife dynamics influence tick abundance and disease risk. Beyond his academic work, Dr. Kwak co-chairs the IUCN Parasite Specialist Group, contributing to global discussions on parasite conservation. Who would want to conserve parasites? I don't know. And zoonotic disease ecology. And so today, I'm so grateful for you. You're just a blessing to work with, Mackenzie, and we're very privileged to be here today. Thank you.
Thank you for having me, and thank you for everyone that's here this morning. Or this afternoon. For Japan time, we are 10 a.m., but for Australia, I think it's about 12:04. So thank you for coming, everyone who attended. So broadly, this is just essentially an information session for the Tick Science Alliance's one of the two bigger projects we're running, and this is the Tick Observatory. And essentially, the kind of impetus for launching this project is that Australia has a very rich diversity of tick species, and undoubtedly a rich diversity of interesting tick-borne microbes, and also interesting tick-borne diseases. And kind of as an ecologist, my perspective perhaps a little more biased than the virologist, or a bit biased away from what the virologists or bacteriologists might feel. I think that broadly, if we understand the diversity and the ecology of our ticks, we understand in large part the diversity of pathogens or the epidemiology of tick-borne diseases, which might be causing damage to humans in Australia. And we have some concept of the ecologies of some of the common species. So things like the northern paralysis tick, Isodes holocyclus, we have a fairly good understanding of its ecology. The southern paralysis tick, Isodes cornuatus, we have a little bit, but not nearly as much as with holocyclus. And there's a few other species like Hemophysalis longicornis, which is one of the cattle ticks, which also causes, say, mammalian meat allergy. And so we understand a bit about the ecology of some of these things. But Australia has about 70 species of ticks. More than just those three bite humans. And so the interest from our perspective is how can we expand our understanding of the ecology, the distribution, and the phenology, so the seasonal activity throughout the year of many of these other tick species, as well as some of the really well-known species, because, say, taking the northern paralysis tick, it occurs all the way from almost Cooktown all the way down into close to Lakes Entrance in Victoria. And that's a very wide distribution. And so we expect that the seasonal activity should vary quite significantly. Because, of course, when you're up in Cooktown, you get a very strong seasonal monsoon, a dry season and a wet season. Whereas when you get down to Lakes Entrance, it's a much more seasonal uh, environment, kind of with multiple different kind of climatic and seasonal factors happening. You can get 4, 5, 6 different kind of seasons depending on which kind of calendar you want to use. So across those distributions, what we know about, say, the paralysis tick in Brisbane doesn't necessarily equate to the ecology and the phenology of the paralysis tick in Lakes Entrance or as far north as Cooktown. So even for the common species, we really want to understand what they're doing within more of a localized area so that we can not only understand their ecology better, but we can start to actually make predictions and recommendations that can be used to control ticks and to limit their populations in hopefully sustainable ways. Um, so the broad idea of this is to try and mobilize as many citizen scientists around Australia as possible to set up essentially their own little study site and to be monitoring those ticks more or less long term. I mean, of course, we're preliminarily asking people to do it for 1 year. And typically for a phenology study, we would be doing about 2 years because you might have a freak hot summer or you might have a freakishly cold winter or a very dry autumn or something like this. Whereas when you have 2 years or 3 years stacked against each other, you get some idea of what the mean of your kind of trends of your, say, ticks or other organisms is. But we're going to start with 1 year, see how it goes. If people like it, we would love it if people would continue for more than a year and would continue it for 5 years if they really wanted to. Um, so broadly, that's kind of what we're hoping. Um, and to kind of give people an idea of the specifics of it more, essentially, we're going to ask citizen scientists to basically pick one spot somewhere in their local area, somewhere that's relatively easy for them to access. But we also want people to find spots. And of course, I'm going to help every individual citizen scientist pick their kind of perfect study site. But we want to make sure people are sampling sites that are ticky. Like, there's no point sampling your local oval because very low chances that you get particularly high diversities of ticks as they're getting mowed kind of every, uh, I don't know, every 2 weeks. And then, of course, the AFL boys are running across them every 2 minutes. But so we want people to kind of be able to find a really nice sampling spot. And these can include kind of areas like local bushlands, um, the heath and scrub along kind of the beach if you're living, especially Sydney way, is very, very ticky. But essentially, we're interested in places where humans overlap with ticks, where the kind of sylvatic ecology of ticks coincides with this kind of, I don't know, synanthropic kind of spillover zones, these kind of halfway points. So these are things like bushland reserves, walking tracks through forests, things like that. And these are also places where you have pretty good wildlife communities because the wildlife are the ones that are really moving the ticks. So these are things like uh, bandicoots and wallabies and wombats and echidnas are all kind of tick taxis essentially through these areas. And so we're interested in places where wildlife and humans interact in habitats that are pretty happy for ticks, basically. Um, and so the idea is that each individual will find, or each citizen scientist will guide you on how to find your perfect site. And then once you've found your perfect site, the concept is to essentially be flagging a set length of area. So this can be, say, 200 to 500 meters, a kilometer if you really want. But I mean, 200 to 500 meters is typically a pretty good one. And then doing it for a set period of time. So for this, basically, we're going to recommend about half an hour. And we want people to be pretty strict on that. Um, and essentially, we're going to be using a technique which is called flagging. And so ticks are um, ectoparasites, which in most cases, not always, but the species we're interested in, basically sit out in the forest and they're waiting for some host animal to go past. And so they'll be using olfactory cues, so they can actually smell the CO2 coming off your breath and they can smell other what we call pheromones, so different chemicals that you're releasing um, which are attractive to them. And they can also feel changes in the air. So as you move past and you displace air, they have little seti, little hairs, sensory hairs, which they can use to feel the change in the movement of air. So this gives them cues as to when to lift up their arms and latch onto you or some animal or something else. So when we do this technique called flagging, essentially we have a large white flag, which every participant will be sent on a pole, and we're basically just waving this across grass or leaf litter across the ground. And as this passes across, ticks mistake this for an animal and they latch on. And so we can very easily, in a somewhat standardized way, be collecting ticks in a safe way. And then on the white flag, we can pick them up with forceps and we can put them into ethanol to preserve them and to use them to basically identify what ticks are in your area and what's their change, what are their changes kind of in abundance throughout the year, or potentially also throughout multiple years. If you have a massive change in your wildlife or your landscape or your forest, this can result in massive changes in your tick populations. And suddenly, an area which had no ticks before can become extremely tick heavy if you have shifts in your wildlife community or your land use. Um, and so we'll essentially send everyone the sampling material. And we're also going to do, when we've kind of when all the citizen scientists kind of have their material, we're going to do a kind of live tick flagging demonstration. We'll just all connect via an iPhone and we'll have a go in the field all together. And I'll find a forest site here in Japan and I'll take my flag out and do some flagging, and you guys can do your flagging, and we'll kind of see what we get and we'll get a feel for the techniques and how we should kind of be doing it. And I'll kind of give you some kind of ticks and tips and tricks for getting ticks. And so hopefully, as we've uh, gone through this, everyone will have a productive site and you'll actually get a really good idea of which species hang around your area. When are the high risk times? Because, of course, that's going to shift depending on where you are, depending on the species, depending on the climate, depending on the location. Um, and through this, hopefully, over multiple years of sampling, we'll build up a really clear picture throughout Australia of which ticks are where, when are they active, what's in the community. Are there some species which compete with others and displace them? Are there some species we never find together because they one species outcompetes the other? And these are the kind of questions we're kind of interested in answering. And through this, we then get an idea of how pathogens or, say, non-pathogenic diseases like tick bite anaphylaxis or alpha-gal syndrome or these kind of tick paralysis, how all these are kind of what their epidemiology is throughout Australia. Yeah. Sharon, do you have anything you'd like to add?
Well, can you explain how you explained to me that there's quite a discrepancy in Australia in tick science in regards to their impact on animals or livestock as opposed to human interaction?
Sure. So Australia is, I mean, the old saying is Australia was built on the back of the sheep, right? And Australia is a huge agricultural nation. In fact, our second largest export is agricultural produce right after minerals. And the third is education, if you can call that an export. And the fourth is tourism. But agriculture is a huge export industry and it has been for a very long time. As Australia, I guess you'd call it industrialized, uh, the major product was agricultural goods, and a huge amount of that has been livestock. So sheep, cattle, and to a lesser extent, things like dairy outside of meat production itself. Um, and because of that, Australia at the government level and at the academic level has always had a very high focus on basically veterinary science and agricultural livestock health. Um, because we have a population of animals which far exceeds our human population. And so the majority of our kind of scientific data on, say, ticks and in fact many other parasites is heavily biased towards animals, particularly domestic animals and livestock. And this has meant that we've had a very limited uh, work done on medical entomology or medical acarology and kind of things like tick-borne diseases. This has changed in recent years. So since I would say probably the turn of the century, maybe the late kind of the 90s onwards, uh, human medical aspects of acarology has increased in popularity, certainly. So these are things like the discovery of Flinders Island spotted fever, which is now the pathogen we call Rickettsia honei, and this was done by the Australian Rickettsial Reference Laboratory. And then, of course, even before that, there was knowledge of other tick-borne infections in Australia, like Queensland tick typhus, Rickettsia australis, which is another spotted fever. Um, and so there's a few things like this, but really, in terms of our understanding of the medical impacts of ticks, it's very much in its infancy. We can kind of compare it to a country which I guess more or less got going in medical acarology kind of about the same time as Australia, which is Japan. And so the very first time that Japanese spotted fever, which is a massive, serious infectious disease and is actually present in Australia as well. Um, this was first reported in Japan in the 90s. And this is about the same time as Australia is discovering things like Flinders Island spotted fever. In that time, though, Japan has a much larger population and a much more robust scientific base. And I mean, consider that Japan has a population of 120 million to Australia's 25, 28 million kind of people. So Japan does have some advantages in that. But in that same time, Japan has done incredible things in terms of detecting, characterizing, and understanding the pathology, epidemiology, and diversity of tick-borne pathogens. So just in the last few years, there's been the detection of things like the Oz virus, which is a new the index case was a fatality, unfortunately, but it's a potentially deadly zoonotic virus. Just here in Hokkaido, where I am, colleagues just across the way found Yezo virus, which is another nasty zoonosis spread by ticks. Of course, a few years before that, they found the SFTS virus, which was originally discovered in China and then was subsequently discovered to have spread to Japan and Korea and has moved its way down south into places like Vietnam. There's been seroprevalence detection in Borneo, Myanmar. So, in terms of even just tick-borne virology, uh, the Japanese have done incredible things, and the Australians have, unfortunately, not kept up quite as well. So we have basically very, very little understanding of any tick-borne viral zoonoses in Australia. Bacteria, understandings of bacterial zoonoses is better, but it's very, very focused on the Rickettsiaceae. So these are the Rickettsia, the spotted fevers, and then Orentia, which is in Australia also called scrub typhus. So we understand a lot about that. There's been detections of things like Ehrlichia canis. There was very big kind of discussion of this when it was detected in the Northern Territory a few years ago. This is also a potentially zoonotic pathogen. So Australia could be doing better than what it's doing, and the hope is that by advancing things like say, understanding the biology of ticks, we provide that kind of key to understand the movement of these pathogens as they're increasingly discovered. And even though I'm here based in Japan, we actually do screen Australian samples. And so we're presently looking for some tick-borne pathogens with some partners from Australia. So we kind of hope to be able to contribute to that as well. And certainly colleagues here at this university are planning trips, research expeditions to come to Australia to gather ticks to screen for tick-borne viruses because they kind of view it as such low-hanging fruit because almost no one's doing it. Um, so there's a big way to go in terms of, say, medical acarology in Australia, but we're kind of trying to bridge the gap, and hopefully projects like this will actually bridge the gap, and we'll get a much better understanding of the pathogens which are making Aussies sick.
Awesome. Does anybody have any questions for Dr. Quack? Just if you're muted, you might just have to unmute yourself. It's
Yeah, any questions at all? It doesn't have to be about the project. It can be broadly tick related or ecology related or something like that. If there's anything that you want to know, please feel free to ask because that's why I'm here. So if there's
Um,
anything you've ever wanted to know, it's a good chance.
I'm just wondering, when you say we do the same site, does it actually have to be the same site or in the same vicinity area?
It
Like,
has to be exactly the same site. It has to be exactly the same path. So the idea of doing that is that uh, between I like one thing that I've kind of, I mean, if you do it, you'll understand. But one thing that I found from even my kind of work here in Japan, especially, is between 5 meters within a single forest, you can have massive differences in the number of ticks. And this can just be that it's slightly shadier in one area, or there's
yeah.
a track in one area that wild pigs and deer are moving across. So the idea of using exactly the same site, keeping everything standardized, same technique, same individual flagging, same site, monthly intervals, all this kind of stuff. And ideally, we want to try and keep the weather somewhat similar. So if you do it, like, I mean, never do it in the rain, but also you probably don't want to do it on a stinking hot 40-degree day. But the idea of doing this all standardized is that we can then compare every month. Whereas if we're doing it between two sites, it's hard to set, or two locations in the same forest, it's hard to differentiate whether the higher number of ticks collected might be a microhabitat aspect of there's just more leaf litter or it's just slightly more humid because it's in a valley versus a seasonality difference, which is triggered by some kind of uh, core ecological and evolutionary kind of driver of when the tick has been selected to be active throughout the year. So we really want to keep it highly standardized so that we really can compare. And the idea of having the multiple sites is not that we're necessarily going to compare between, say, a site in Victoria and a site in Queensland, but we want to understand across those sites what is happening potentially even with the same species. So if we have someone in East Gippsland and we have someone in Sydney and we have someone in Cairns, it's going to be very interesting because they're probably all going to get, say, Isotes holocyclus, the northern paralysis tick, but the phenology is going to be potentially radically different depending on the climate that they're sampling in. And by getting those kind of smaller scale observations, we can get a much better idea of the diversity of phenology and activity of a single important tick species like Holocyclus across quite a wide range of uh, climates and habitats.
So I'm on the edge of a national park, which is quite
Hmm.
well used. So if I did the left side of the road, which is the river side of the road, 1 month, and then the national park side of the road the next coming back, no, I
No,
would
no,
just do the
never. You
one
have to
side of the
do,
road.
yeah, pick your side
Right.
and do it. Sometimes I've had this happen as well. Sometimes your site will be massively disturbed in the middle of a study. I had
Right.
this when I was in
Yeah.
Hokkaido and I had a beautiful deer track along and then they turned it into a road the next month. And I was getting loads and loads of ticks the first month. And so we actually threw that site out of our study because we just couldn't get the second month of data because it was flat and there was basically nothing there. Like it was just dirt.
Okay.
So in those cases, we might adjust it. So we might switch to the other side of the road because it's still kind of broadly very, very similar. Especially if it's like a track, a small track, then doing one side or the other side actually doesn't matter. But if it's sort of like I'm going to go across the valley to do a different path or a different road, that's kind of a no-no. But ideally, as much as possible, we want to keep it. Ideally, if it's a road, we want to keep it probably the same side of the road where possible. Or the alternative is to do both sides of the road and do 15 minutes along one side and
Okay.
15 minutes along the other side.
Yeah.
But
Because it's
keep
a
it to
sand
one
track.
side.
Yeah.
Yeah, right. So
Sand
just
track.
keep it to one site.
And then up
Um,
back and call it one site.
Yeah, when I'm doing my sites, kind of my long-term study sites in one of my study islands, uh, I'll do both sides of the track.
Okay. Right. That's good. Thank you.
Very welcome.
And have all the ticks in Australia been described? Or are you expecting
No,
novel
no,
species?
no, no. We may find new species. I think it's less likely. The new species we're finding now is predominantly nest-dwelling species, and this is just because they're harder to detect, and you really have to go into the nest of the or the burrow or the den of the animal and detect them. But there is a good chance that we may detect some new species as well. And even in like Japan, where the ticks have been heavily worked before I got here, I've got like 5 new tick species sitting on my desk waiting to name. So it wouldn't shock me if we're finding this many in a tiny country like Japan. Australia would have loads and loads and loads. And we're kind of hoping to try and clean up some of the Australian ticks, kind of their taxonomy as well. Jennifer, did you have a question?
Yes, thank you. I was just going to ask about the flexibility around, is there a time restraint? So once a month, does it have to be
It doesn't have to be
within
exactly the same day.
that not the same day?
No,
So
no, no. So if you say do the 15th, it doesn't have to be the 15th every month. And
sure.
the idea of this is one, we want to be kind of flexible for people. And two, we want to be realistic because the kind of conditions are not going to be perfect for flagging on the 15th of every month. And so we try and ask that, say, everybody does it, say, within the middle 2 weeks of the month. And
Hmm.
you can pick any day within those middle 2 weeks of the month that's suitable. And so for this, we specifically want to pick days. Uh, One, we would never do it when it's raining. It's not good to do it when it's very high wind. It's best to do it if it's rained 2 or 3 days beforehand, because then the leaf litter has had a chance to dry out. If you do it the day after a big rain, your flag is just going to totally soak up all the water and it's going to become totally ineffective. So you want basically it to be dry, but not hot and windy. And so within kind of the middle 2 weeks of the month, pick any day that suits you and any day that's kind of climatically, weather-wise, fairly suitable.
Hmm.
And that's fine.
Okay, thank you.
And are those conditions documented so that you can do the conditions
Yeah, so we're gonna
themselves
we're gonna
affect?
get everyone to record the date
Yeah.
of when they did the flagging, and we can actually use the local weather station data to figure out what the weather and temperature and all this kind of maximum daily temperature, minimum daily temperature, all this kind of stuff was. And we can feed this into models kind of later to try and understand how temperature is affecting some of these species as well.
And one of the things you told me was that like sometimes when you're flagging once a month, you're going to find nothing, and that's also
Yeah,
very important data.
yeah, absolutely. So negative data is still data, and it's kind of like you can't have light without darkness, right? So the negative data contrast the positive data, so hits of actually getting ticks. And actually, throughout the year, we expect that some months you will get nothing. And that's perfectly fine and that's really good. And it's not because you're a bad flagger or anything. It's just because some species go almost totally dormant, kind of. We would call it kind of diapause. So some species will basically become dormant through parts of the year. And we see this very commonly in most tick species. You almost never find a species which is active all year. You find that they come in peaks and you have kind of peaks and troughs in activity. And by understanding when the troughs are, we understand when the safe periods for people to be out in the forest or out in the bush, out in the scrub, whatever, and when are the high risk periods. So yeah, please don't be disheartened. Lots of my sampling sites in the middle of summer, I get nothing in lots of my sampling sites, especially here in Hokkaido. We have species which have very, very distinct phenologies. We kind of call it almost like an Isodes spring peak. So one of the genera we have here, the medically important, the Lyme vector here is Isodes persulcatus and Isodes pavlovskii, and both of them have this funny spring peak. So from about late March through to late May, we have this beautiful peak in abundance, and then they immediately drop down, and we never see them again for another 9 months, 10 months until they come back kind of the next year. And they're just totally absent. They're in the leaf litter, they're not interested in biting. We might occasionally find some larvae, but the adults and the nymphs, we basically only see them for 2 or 3 months of the year, and then they're totally absent the rest of the time. We think many of them are probably in rodent burrows. And so they're acting as nidicolous nest-dwelling species for part of their life cycle, and then they're coming out as exophilic, non-nidicolous field ticks for their nymphal and adult stages when they're going to be encountering humans and deer and raccoon dogs and things like that. So we do expect you to have periods where you find nothing. You may also, as you get better at spotting the ticks, because as you look at them more, you'll start to pick different species probably. You broadly get this is species one, this is species two. You may not know exactly what it is. But as you do that, you might find, I mean, certainly I find in my sampling sites that throughout the year, I've been doing it long enough that I can predict, I know exactly which species I'm going to meet which month. And so you'll be doing it and you say, oh, there's a lot of kind of black looking ticks this month. Oh, 2 months later, oh, they're replaced with these large brown ticks. And so these are often different species or different life stages coming in and out of activity throughout the year. So I think, probably get some interesting surprises of what you're finding throughout your sampling period and within your sampling site.
And are there safety policies? So I know that Lyme Disease Association will supply all supplies, like that will be provided. Is there any other instructions around
Yeah,
safety?
so obviously we want people to dress appropriately. I do occasionally do flagging in my flip-flops. You really shouldn't do that. It's very naughty. And I only do it when I'm on a concrete road, kind of an asphalt road, and I never step on the leaf litter ever, and I just reach my arm across. But you should never, ever, ever do that. So
Thanks.
fortunately, I've never been bitten by a tick in 15 years of studying ticks ever,
Oh,
even though I spend a huge amount of time in the forest. But partially that's because I'll never ever sit down in the forest. I'm always standing and I always wear knee-high gumboots. So if you can bear it, especially in these sweaty summer times, I highly recommend big gumboots. They're fantastic. Even when I've gone hiking kind of on like field trip expeditions into Southeast Asia, gumboots is always my shoe wear of choice. Everybody else has got these very expensive kind of laced up special hiking boots, and they all get wrecked and they all get full of water. Gum boots are fantastic. They'll keep the ticks off. You never have to tie them, um, and
You can
they'll never
turn them
fill up with
upside down,
water.
drain the water out.
That's it. If you go knee high in the
Yeah.
water, you just tip them out, you're done. If you step in with expensive hiking shoes, they'll be soaked like a sponge for the next 3 days. So really appropriate field gear is critical. And so for that, yeah, I'm kind of a great preacher of the gumboot religion, but gumboots are fantastic if you can get them. Um, we highly recommend wearing long sleeve and long pants when you're out in the field. And if you want, you can put a rubber band in and you can kind of use that to attach kind of the leggings and the arms to your wrist, just so nobody ends up crawling up and making a home in your armpit or something. So that's kind of quite important. We also highly recommend regular checks. So while you're flagging, just check your body every couple minutes. Just have a little look around. I'm always doing it when I'm in the forest. Just check, and sometimes you find someone crawling up your leg, and you can just remove them. So regular body checks, protective equipment. We also recommend repellents. So DEET is a very good one to use. It's not necessarily my favorite, but the other ones that you can get, say like permethrin, is a better one because it's actually a touch acaricide. So it kills the ticks when they come into contact with it. It not only repels them, but it also kills them. This is a harder one to get. I don't think you'd be able to get it. But DEET, DEET, you'll see it on lots of things like AeroGuard stuff. This is a good one to use and give yourself a really good spray. You don't need to spray your head. The ticks are not going to be on there. But especially if you're doing, especially your pant legs and your sleeves, this is a really good place to spray. But you don't need to like bathe in the stuff. But just give yourself kind of a good spray so that you're adequately repelled to mosquitoes and ticks. And you'll probably be happy because tick habitat is often quite good mosquito habitat at the same time because there's lots of wildlife and there's lots of micro habitats for mosquitoes to shelter in as well. So you protect yourself from the horse flies and the mosquitoes and the other nasty biting things. Um, we're also going to send everyone forceps. So if people, I mean, these are to collect the ticks off the flags. But additionally to that, you can use them if you get a tick on you, you can pull it off with forceps, and we can give you instructions on how to do that as well. Is there anything I missed, Sharon?
I don't think so.
Okay. But yeah, for this, we really don't want any of you contracting any tick-borne diseases.
What if you
So we
already have
really,
one?
Oh, you already have one.
well, we don't want you to get another one.
Yeah,
If
the
if
sample
you've already
site.
had one, you may have antibodies for it. But if you haven't, but that doesn't mean you won't get other ones. Like I've had flea-borne typhus, and so I probably will never get flea-borne typhus again. I've got very good protective antibodies for that. So I'm kind of immune to some extent. So for some, if you've had, say, Queensland tick typhus, you may actually have some, so there's Rickettsia australis or Flinders Island spotted fever, these kind of things. You probably already have antibodies for it. Of course, the antibody titer will drop with time. Fortunately, a natural infection is much, much, much more long-lasting typically than a vaccination. So if you've had a natural, I mean, I don't think there are rickettsial vaccines available in Australia, so that's not really a point. But if you have had something like a Queensland tick typhus, you should have some protective immunity, unless you had it 40 years ago, in which case your antibody titer might be very, very low. Kind of your memory cells will be quite low. But just because you've had one tick-borne disease doesn't mean you don't get another one. And there are some very, very nasty ones out there. So we don't want anyone getting anything, basically. So we want to be extra, extra, extra tick safe because it's very unfortunate and very difficult to study ticks if you suddenly get ill from a tick and you have to drop out. That's bad for you. It's bad for the study. We want to keep everyone safe.
And I think Stephen has a question.
I was, as Sharon said earlier, involved in one of the first tick diseases support groups down on Sydney Northern Beaches.
Mm-hmm.
And as part of the group, we had a retired entomologist who used to collect ticks for the tick diseases research centre.
And that would have been at Westmead, I guess.
Royal North Shore, I think.
Yeah. Okay.
Dr. Hudson sort of headed up, Benny Hudson headed it up.
Mm-hmm.
And his role in the support group was he had a sheet about 2 meters square.
Mm-hmm. And he'd drag it across the leaf litter?
He used to sleep with it the night before. So it
had his smell.
Yeah.
So for this, we recommend that you do not sleep with the sheet because some of you may smell better to the ticks than others, and we don't want to bias our data very much.
Okay.
So we want to keep it as standardized as possible, and some of you may be very, very delicious.
Rob, you're funny.
Sampling, the site sampling thing that a place that I'm interested in, you don't even need to go outside to collect the ticks.
Oh, even better. So you'll have hundreds.
Yeah, so Sunshine Coast hinterland, Mullaney.
Very, very Isodes holocyclus heavy.
So yes.
I think you'll also get some Hemophysalis, maybe Bancrofti, and we'll see. Longicornis should probably be there too. Stephen, did you have a question?
Yeah, sorry, I was trying to figure out how to unmute. Um,
I
so
have the same problem all
um,
the time.
is it better to like have sunny day and then like rainy day, or you want the same kind of conditions all the time?
Uh, so it doesn't matter hugely what the temperature or whatever was like necessarily the, on the day exactly, because we expect that the temperature should shift throughout the year, right? Like you'll have warm days and like you'll have a warmer season and a cooler season depending on what part of the year you're in and where you are in Australia. Um, The only thing we want to try and do is to try and not sample on super wet days, super dry days, or super windy days. But we accept that within your sampling site, your climate will change throughout the year, and that's kind of part of it. And we also expect that to be kind of one of the critical cues which will be triggering your tick activity. But you don't need to be too pedantic about it must have rained 3 days before I sample, and it must be a sunny day on the day I sample. When we are sampling, though, my recommendation is always to sample in the morning if you have a chance. Don't sample at 8 a.m. because there'll probably still be a bit of dew on the leaves and stuff, and that'll start wetting your towel. But being a tick biologist is actually, in my opinion, the best kind of biology because unlike the bird people, which have to wake up at 4 a.m. to listen to the morning bird call and identify all the birds
You could
in the
sleep
morning
in.
chorus, tick people, we sleep in, you wake up at 9, you go to the field at 10, you do your sampling, you're done by midday kind of thing. So ideally, I highly recommend doing it kind of in the morning before lunchtime, but maybe after about 8 a.m. or 9 a.m. And the idea is that this is a really nice temperature zone for the ticks. Typically, it's quite a comfortable temperature, so it's rarely above 30 before midday in most places. And it's typically uh, not had time for if there was morning dew for it all to totally dry out. So the ambient humidity is often a little bit higher in the morning before lunchtime. So at least when I'm sampling, I'm always trying to do it. If it depends on the site a bit and the location, the island or the wherever I'm doing it. But typically a really nice time to sample is about 10 a.m., 9 or 10 a.m. in the morning.
And Chris, you had a question?
Yeah, just on site disturbance. A lot of the areas I've been working in, there might
You've
be
got an
on
echo.
grazing.
Hmm.
Sorry.
There's an echo, but do you understand? Can you hear it, Mackenzie?
Yeah,
Okay.
yeah, I can hear it. Keep going,
Yeah,
Chris.
yeah. So the sites might be subject to grazing. They might also get burnt at various times. They might get slashed. Is that going to worry too much, or that's part
That
of that cycle?
that worries me a little bit. So if they're getting slashed, slashing will change your microhabitat drastically and will also be shifting things like the ambient humidity within your microhabitat. If they're also getting burnt, which I know I don't know what they call them, the Department of Environment or the Department of something services loves burning Australia. If your site gets burnt, uh, that's really, really bad. And your ticks will
Only
not
low
recover for a
intensity
while.
burning. Yeah, it's only low intensity burning, and it's
Okay.
just part of ecological management of sites.
Okay.
So, so
Of course, it's site dependent. And of course, if you're in a grassland, you need regular burning, whereas if you're in a wet sclerophyll forest, you should have almost no burning, right? So don't get me wrong, I'm not saying all burning is terrible, but I would say that at least what would we say? The standard for Australia is probably that a lot of Australia is getting burnt way, way too frequently, and the interfire interval is not being extended enough, which is driving the selection for highly flammable pyrophilic flora and displacing a lot of these more sensitive. So, I mean, you think of like, I don't know where you are, but at least in the Dandenong Ranges, I've watched a lot of it, of where they do these regular, incredibly stupid burns of wet sclerophyll forest, and you get this displacement of sensitive species like Dicksonia, the tree ferns, and they get replaced with all these really, really super flammable, lots of POA, lots of myrtaceous species. And of course, it makes the whole environment much more flammable. But that's kind of beside the point. What I'm trying to say, though, we can argue ecologically about which kind of uh, floral, uh, say bioregion or whatever, how it should be fire managed. Some of them definitely need fire. What I'm saying though is if your habitat has a fire through it, it does have this kind of post-fire recovery period, which sometimes takes quite a few years for fauna to bounce back. Some fauna, like especially some small mammals, really love that interfire period, and some have very specific intervals within that interfire period they want. So things like smoky mouse, and New Holland mouse, the pukilla, have kind of sensitive time periods which they like. But many species will be kind of locally extirpated for a period of 1 or 2 or 3 years before they start to move back in. And so things like, say, echidnas, they can come back in very quickly, but resting areas maybe get burnt up, these kind of old logs and stuff. And in some cases, the ticks are actually living a lot of their life in the nest. And so when you burn up the nest, you burn up the ticks, and then it takes a long time. Even if the echidnas return, the ticks take a few years to come back after that. And so when we have burning periods, often it's quite, I mean, you can think of this as good or bad, but it can be quite detrimental to your tick population. So if you have a study site that gets burnt halfway through the study, we definitely want you to keep sampling because it's interesting data about what happens to ticks if there's a burn. So it's not necessarily going to tell us a huge amount about phenology because the site has been totally altered, but it would be really interesting data to be able to see what happens to tick communities after fire. And if we have the before and after, that's fantastic. So ideally, we'd prefer sites that don't get burnt. If they do get burnt, though, we'll do a totally different, basically kind of observational experiment of looking at what happens to tick communities before and after fire. So it's not like it's the end of the world or anything. But if we can select an area that's not going to be burnt, that would be preferable. Although if you can't help it, I mean, we take the data that we can take, and it's an interesting kind of quasi-experiment anyway, and we'll write it up as something else.
Yeah, well, I'm in Western Queensland.
Okay.
So, the sites are often prone to grazing, and the grazing there for a and then on for 12 months. But some of the sites might have, it's just mosaic burning, so there might be a corner of the site might get burned or part of it, and it's only for low intensity, so you're still leaving a lot of vegetation, you're leaving all the ground timber.
Mhm.
So, and we know from experience that the ticks are still there even after burning.
Okay.
But
So it's really, really low intensity burning.
it's very low intensity burning. Really just for ecological recovery.
Hmm. I mean, it's
So-
interesting. It would be if you want to sample that kind of habitat, as long as we kind of from the beginning have some idea of what the regime of, say, grazing and burning will be, and it might go over a few years, then I think it's a really nice data set. And it's maybe not perfectly pertinent to exactly the question we were originally asking in the kind of tick observatory stuff, but I think it's a really nice complementary question of looking at what happens when we do different grazing techniques or different burning techniques on our tick communities, and can we manage them in a kind of more sustainable way through land management rather than through nuking everything with masses of acaricides, which disrupts soil communities and mesofauna and all this kind of stuff. So
Yeah,
if that's the condition, let's do it.
It might not be disturbed at all, or it might go through a massive drought, which will alter things again. Just trying to find a sort of science consistent, but it may be subject to a lot of environmental change.
I mean, I don't think it's that terrible. I think we can work with it. I think it's okay. And I think it may be, I really think it may be worth doing something as kind of a spin-off study of looking at a tick community, if possible, over a long period, say 3, 5 years, something like this, and seeing what happens to it year on, year out as, oh, it's slashed this year and the other 3 years it wasn't slashed. And we can kind of run a model and see whether that has any kind of significance. Or kind of like how much effect that has on our kind of, say, abundance or community diversity of ticks. So for me, I mean, I really like phenology. I like knowing where ticks are. I know when they're like knowing when they're active. But actually, in my heart, my core interest is understanding how land use change shapes parasite communities. So something like slashing or low intensity burning is, for me, actually quite a much more kind of intellectually stimulating question to address and to kind of want to understand than just what the phenology is. So I think I think it's really a cool system that you've got there, and it would be really fun to see how the ticks are shifting across months and across years.
No worries.
Any other questions?
Can I just want to share, like because I've had tick anaphylaxis. So I've actually got insect shield overalls. So I don't know if anyone else is interested in those, but I actually got them on eBay. So they're like $400 if you buy them directly from the company, but I got them on eBay for only $65. So I still put cuffs on the wrists and tuck my socks in, but I know the bush that I'm going into, I won't be doing them without full protection. So just always like to share that because I found them really good. And then you can also update them and you can buy the permethrin spray, and I spray them probably every 12 months. Just to share.
Thank you.
That's fantastic. No, I mean, the more we're tick safe, really, the better everybody's life will be.
I have a question in regards to identification of us as tick scientists. I would suspect that some of us might encounter interesting approaches from people who want to know what we're doing they see us on a regular basis entering property that we might not know is privately owned or from a government department, for instance. Will we be given an identification card or something that we can flash and say, hey, I'm part of the scientist?
I think probably before the sites are set, when we're in the selection process, if they're going to be reserves or something like this, we're going to talk to whoever the managing authority is, whether that's state government for state parks or council for local reserves, and we're going to get the tick of approval. And so hopefully we're going to ask everyone. We'll have a letter from the council to authorize it or to at least facilitate it. And then if other land managers are in there and they're sort of asked, what are you doing? You can at least present that so that there's some kind of authorization. But I mean, at the end of the day, Tick Science Alliance is not the managing authority of any of these parks. We're going to be guests, researching them and studying them. So we'll make sure that when we find sites that say I and the citizen scientists like, we'll talk to council and explain what the process is, what the benefits are to the local community, and why it would be in everybody's interest for this kind of study to go ahead. So we'll make sure that we're documented, authorized, and kind of approved to be doing this kind of work. Yeah.
I didn't realize that we'd be doing it that officially, but that's tremendous.
Thanks, Rob.
That will certainly give the results a lot more credence, won't it, being official like that?
Yeah, definitely. And I mean, we wanna play friendly with everybody involved, all stakeholders. We're definitely not going to be kind of rogue.
Community.
Yeah. And as well as this, at the end of every 12-month period, we're going to send out basically a report on what's been found through the preceding 12 months. And for each individual citizen scientist, we'll go through and identify, we'll give you a personalized report on what species have been detected in your area so that you can have a better understanding of what the species are involved, potentially what pathogens they're associated with, or what tick-borne diseases they're associated with, so that you have a much better idea of what's happening in your local space.
Awesome.
Any other questions?
Yeah.
Yeah, could I ask how many people are part of this study at the moment, and is it over a number of countries or
So
and how long
the
has it been
study is
going for?
so this is a new study. We're only starting this now. We've got, I mean, it's not exactly part of this study. I have long-term study sites in Japan, and then I have some in Singapore. And this is just from my own kind of research on ticks in that area. For Australia, this is kind of the launch of this program. And the idea is that we'll start with this is sort of the pilot phase. And so the first year we're aiming for 10 citizen scientists, but the aim is to eventually upscale this until hopefully we have more than 100, which would be really nice and would give us a much clearer view of what's happening across Australia. So this is the very beginning. I've kind of run projects like this kind of in Singapore, but mostly, a bit in Singapore, but especially in Japan. But a lot of the sampling is done by me or by colleagues of these kind of continuous long-term study sites. So this is the first time trying it with a whole bunch of citizen scientists. So we have no idea how it's going to go. We hope it goes well. I think it's not a terribly hard thing to do. It's just about putting in the time once a month to kind of do it. Um, but yeah, the idea is for this to continue indefinitely, hopefully for many decades to come. Even when I'm pushing up daisies and turn back to dust myself, I hope that someone else will have taken over doing the identifications and doing the reports each year and publishing data and stuff on this. So the idea is for it to be quite a long-term data set. And also this is very useful if we have a very long-term data set because we can see, especially in some locations, how ticks have changed over, say, a decade. Or if we then go back in 20 years' time and sample exactly the same location, are there shifts in our tick community or tick abundance? What does climate change do to it? What does land use change do to it? In some cases in the outback, what does salinity do to our tick communities as it shapes the vegetation community? So yeah, so far I think I mean, Sharon, we're aiming for 10 people to start with, and then if that all goes well, we'll just keep expanding. That's kind of the plan. Yeah. Jennifer.
Yes. May I ask, are
Hmm.
you collaborating or liaising with any research facilities in Australia on this, or is this completely independent to anything happening?
Mostly independent. We have one colleague who's associated from Flinders University.
Mhm.
Um, but broadly, this is Hokkaido University slash Tick Science Alliance
Right.
kind of thing. Yeah.
Yep. Okay.
We've actually learned that it's best to be independent of Australian academia in the short term.
Okay.
I don't
But
think they're not all like,
no,
they're
they're
not bad
not.
people. They're perfectly nice. Some of them are perfectly nice, not all of them. I have people as well that I'm not a gigantic fan of within the Australian community. But by and large, I found that the Australian community, at least to me, have always been very, very polite, quite collegial, very kind. They ask me to review their students' theses sometimes. I peer review lots of their papers. So like there's no animosity
Yep.
whatsoever, and we're on very good terms. Um, but the question, I think our question was, does including more tick researchers from Australia enhance the study in any way? Like if we're going to add more collaborators, is it going to enhance it in some way? And broadly, the uh, what we need out of this is not necessarily more people to do. Like I can run models perfectly fine. I've been doing it for 10 years. I can identify ticks. I've been doing it for decades, for more than a decade. So what they would bring, I'm not exactly sure because we have the kind of capacity to run it more or less ourselves. So adding more people, all it does is add uh more people in a discussion, which
Yeah.
sometimes means the discussion lasts 10 times longer.
Too many cooks.
Yes, 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 exactly.
Yeah.
Yeah.
And I should alter it. It's not really the academics so much. It's whoever, it's regulatory bodies and people who influence put pressure on their careers. And I've witnessed people partnering with us who've had their careers harmed. So, if we can progress science and keep it independent and protect Australian scientists, that's also, something that's important to us. Because I don't, the people I've worked with, I've seen, I know, sorry, I'm going to probably try here, but I've seen terrible harm happen. And it's not each other necessarily. And like Mackenzie said, it's not the individuals, it's the system. So, I'm really excited to be able to partner with people like you guys that you just like the salt of the earth. Like when I was talking to some of the people when I've been reaching out, because this is not my area of expertise, as you might imagine. And I'm now deathly allergic to the bush and walking anywhere. But anyway, it's been so amazing and a privilege to talk to people that just want to progress, link arms. They may not be an academic, but they can, all our contributions are really valuable. And there's a lot of people really interested in progressing citizen science. And I'm like, I need to ask your help to apply to another one, Mackenzie, because they asked me all
Mhm.
these questions I didn't know how to answer. But there's people looking to make the Australian bush safer. And that
Hmm.
makes me really proud, like it's just good people.
Well, I think at least from what we're finding in Japan and at least the kind of stuff we're testing here, there are ways to manage landscapes to make them more uh, tick safe. And also, I think a huge part of it is just minimizing risk. And this
Yeah.
is kind of 90%, this is 90% cheaper than like treating people once they're sick. Like once you have
Exactly.
someone in a hospital or once you're sending someone to a clinic, you're losing work hours, so you're losing economic productivity. They have morbidity or in some cases mortality, and so it's a huge loss. So if we can get it before anybody gets sick, that's fantastic. And it's hardly noticeable because, of course, you see a lack of tick-borne disease cases and you say, oh, we're doing fine on ticks. But a lot of it can be through
Mm. And public
mitigating
health
risks rather
awareness,
than managing
mainstream.
disease.
Yeah.
Yeah,
Yeah,
yeah, yeah, yeah.
yeah.
So
And
the
the
hope
data
is
will
to
help
eventually
progress
be
that, won't it,
applying,
Jennifer? Yeah,
yeah.
Yes,
So we
exactly.
want to apply some of those lessons.
yeah.
Yeah, those of us who've had personal experience trying to broadly educate people without, public health backup.
Mm.
So, yeah.
It was interesting. The other thing that experience I had in talking to people about this project in the last 2 or 3 weeks is Gideon, which is an international data organization. I don't,
Hmm.
Mackenzie might explain it better than me, but I had a meeting with the managing director, and they voted this project on their Facebook page this week. It's like people can link arms in their area of expertise and progress science in whatever way they have influence and connections. And I'm really grateful. And it was just inspiring to me to meet these people that just want to support work like this. So, um, yeah, we'll be able to share it if you have other people you want to share this recording with, and if it resonates with them, great. Again, we just want people for whom this is right for. And I'm going to, if you have any people I should call, I'd be calling again this week, talking to people. Um, and I'm happy to reach out on your behalf, or please just introduce me. Anything else, Mackenzie?
No, any other questions from anyone?
So the next
Broadly
steps, if
okay.
you'd be interested, do you want just an email, Mackenzie, or do you want the application?
Yeah,
We do
so
have an online
no,
application.
yeah. So if you want to send us an email, we can send you the application documents, and then if you're like quite serious about it, we'll send you kits of which will include all the sampling materials. And then from May, we're going to
Yeah,
do a practice,
in the
kind of a
field and start the
Zoom
project. And
group, Zoom practice flagging in the field altogether of anybody that wants to kind of be involved. By then, you will have your kits in your hot little hands, and we'll all go out into the field together in our various parts of Australia, and we'll have a go at flagging some ticks, which should be quite fun. And kind of getting a feel for technique and the whole kind of process of what to do with your samples, how frequently to check your flags, how you should be flagging, all this kind of stuff, and also checking out the sites and seeing if the site is suitable. Yeah. So it should be a lot of fun. I always love going. I mean, I'm probably a bit biased, but I love going into the field and catching ticks. It's so much fun. It's like opening Christmas presents. You never know what's going to end up on your flag, and there can be, I don't know, half a dozen species in some sites. So you can get quite a really rich diversity of different ticks.
you're
I
obviously
don't
not a
think
Lyme
we'll.
patient.
No, well, I'm not a yeah.
Oh, that's too funny.
Ticks never bite me, so I'm all right.
Oh, that's a good affirmation. I'm going to use that one.
King of the ticks.
Yeah, master of politics. That's cool. So, and in those packs, there'll be a Ziploc bag, and there'll be educational materials and everything that we've spoken about. But also, we'll provide the tweezers, the tick removals, and
Yep.
also the
Flags, flagging poles, collection tubes with ethanol in them. So it
and
should
the
be quite easy.
DEET. I can't
Labels,
think of the name
DEET,
for it. Yeah, we'll
yeah,
supply that as well.
yeah, repellent.
Sorry, just once we have them in our tubes, where do they go? Like we have to
Ah,
get them
good,
to you.
good question. No, no, no. So
Lucky me.
Or to
yeah,
you.
so once they're in the tubes, we ask. So basically, we have preservative alcohol essentially in the tubes, and so this is gonna basically we put the ticks in live. This kills them and also fixes them, preserves them at the same time. So then once and we put a little paper label kind of like this written in pencil will go into the tube as well with a little bit of information. This is going to include your ID, your collector ID, citizen science ID, the date that you did it, and probably your name as well. just to be totally sure in case we can't read the numbers or can't read the name. Um, and this is then going to go into a little Ziploc bag, and we ask people to put it in their freezer. And the idea of doing this is that the ethanol will preserve the DNA somewhat well, but keeping it frozen in sub-zero conditions will preserve the DNA even better. And the idea of this is that uh, later on, we would love to be able to potentially look for pathogens in the samples. And if we can take a subset, if people have been collecting ticks all around the country and we have the DNA preserved very well, we can start screening for different zoonotic or pathogenic bacteria or protozoa. Unfortunately, viruses, not all of them, but the majority of them, of the viruses we care about are RNA viruses. And RNA does not preserve well in ethanol at all. So we can't screen most viruses, but we can screen protozoa and bacteria. So we can look for things like rickettsias and borrelias and ehrlichias, anaplasmas. We can also look for protozoa, so like babesia, Thaleria, hepatozoon, these kind of things. So we ask that people put in the Ziploc bag. It's not going to be infectious. It's not going to be dirty. Uh, Ethanol is a very good sterilizer of many things. Um, and then just put it in the back of your freezer. And then every month, add the next month's tube. And then every 6 months, we ask that they get mailed back to Tick Science Alliance, which I think is going to be
Me.
Sharon.
Yeah.
Yeah. And so then probably every 6 months or once a year, they'll be sent to my laboratory here in Japan. I'll go through, identify them, and then send everybody the results of that year's sampling in the final year report.
Our last conversation, you thought you might want to do it quarterly, send the
Yeah,
tubes in.
potentially quarterly or 6 monthly.
Yeah. Okay.
I guess we'll figure out what we want to do and how people are going with it all. Yeah. So, yeah. But they're
Good.
very small tubes. I mean, they're like, I've got one. Yeah, like this. Kind of little tubes. Yeah, it's full of
Wow.
these are sea snake ticks, Amblyomynitidum larvae that we reared out in the lab. But this is how big the tube is going to be, basically. And so then it's literally just a matter of putting ticks in, putting it in the freezer, and then sending it to us.
Frozen?
And
Do they need to?
no, it doesn't need to be frozen. The point of the keeping it in the freezer is just to try and keep it sub-zero as long as possible. Just because it warms up to room temperature doesn't mean that it's going to suddenly all be ruined.
Yeah.
The idea is just that if it's sitting at room temperature in ethanol, the DNA will degrade much faster. But I mean, once you bring it up to room temperature, the DNA will be slowly declining in quality. But 1 or 2 days at room temperature, a week at room temperature is not going to change the world in any meaningful way. But 6 months or 2 years of sitting at room temperature can cause significant degradation. So we want to try and keep it on sub-zero kind of ice as long as possible. But in between, it's just it'll be slowly declining, but a few days at room temperature is no problem.
And Mackenzie, I think the answer to the question in regards to postal requirements for shipping a tube with ethanol or customs, there's no restrictions, no issues
No,
there.
I'll also give you documents to do that. Within Australia, there's no conditions. We basically ask that people don't say it's
Takes.
ethanol. You can say it's preservative or something like that. If you need to, you can also use the chemical formula for it, which is, what would that be? C2H4OH, I think is ethanol. Nobody knows what that is. I mean, nobody, post office is going to recognize that this is ethanol kind of thing. So we want to be a little bit, we never say, I mean, they're never live or anything. Basically, normally we just say scientific specimens or museum specimens or something like this, and that's normally quite fine. But don't go in and say, I want to send flammable alcohol ticks through the post, or people will like lose their cool very quickly.
Biohazard label. No.
Yeah, all this kind of stuff. So no biohazards, no flammable, none of this kind of stuff. And it's within a 2 milliliter tube, so it's really quite safe. Yeah. But scientific specimens is perfectly fine. And if people ask and say, what kind of scientific specimens? And you say invertebrates or arthropods or something, and if they want to go even further, you can say they're mites, which ticks are technically giant mites.
Mm.
Yeah. But we try and minimize the alarm as much as possible.
Fair enough. Mm.
And the email that we communicate with is the one that Sharon's been working with us?
Yep.
Yep.
And so Sharon is broadly the point of contact for all of this. Of course, I'll help everybody with their individual sites and sampling. And if you have any kind of on the scientific side, if you have any problems or doubts or questions or concerns, you can always contact me. I'm very, very happy to chat about it as long as you want. That's totally fine. But in terms of logistics, Sharon is the master of logistics and management and all of this sort of stuff. So I always defer to her in all of those matters.
And you'll see another name, Venkatesh, is a partner. He's working with me behind the scenes. Very high detailed man.
Detail oriented, yeah.
Yeah, he double checks all my work, which I appreciate. So he'll be managing sending out the kits and supplies and replenishing any supplies if you need extra tick repellent or anything. So, but mostly I do most of the communications. So we're very blessed to have spent this little time with you. Thank you very much for your interest. Just let us know if you want to progress, and we'll set up the logistics and the partnership, and we can go from there. So feel free to spread the word.
Yeah, please tell anybody and everyone. The more citizen scientists, the better. We're aiming for 10 for the first year, but if we can get 20, 30, 50, 100, we'll very happily
expand.
We'd love to have as many citizen scientists involved in this as possible. And the more we have, the better, clearer picture we'll have of what's happening with ticks around the whole country. And that's kind of the overall aim. So tell everyone, tell your friends, tell your neighbors, tell their dog, get everyone involved.
I mean, it's really important work, Mackenzie. We're so grateful to you because
Well,
it's
I mean,
not
I'm doing something very small, which is just identifying the ticks and running the models and analyzing the data.
agreed.
But like the citizen scientists are the ones who will like be in the field, finding ticks, understanding their local area. So those are the people that really make this project run. They're the kind of
You're right.
meat and potatoes of this.
Can't do
So
none of
we're
it
very
without
happy
all
for
of
anyone
us.
that wants to be involved.
That's right. So thank you for being here and for your time this morning. I will send this recording out to you, everybody who's here, but also everybody who expressed interest. And then I'll be listening to what Mackenzie advises me to do next. And you'll all know when I know. Thank you so very much. We very appreciate it. Thanks for being here.
Thank you, everyone, for coming. Thank you for
Bye, everyone.
sacrificing your sunny days.
Yes.
Thank you to you both.
Thank
Bye-bye.
you.
All right,
Thank you so
hooroo.
much. Cheers.
Bye.